Within a sealed 5-gallon plastic container situated on your rear patio, adjacent to a fractured ceramic planter and a sack of pine bark mulch, something truly remarkable is transpiring at this very moment. Every 48 hours, the living organisms contained within that vessel experience a twofold increase in their numbers. Furthermore, the valuable output generated by these microscopic lives, achieved with the modest investment of a single organic potato and a tablespoon of sea salt, possesses a molecular structure identical to a category of plant growth enhancers that the United States Department of Agriculture dedicated 40 years and over 500 pounds of bee-collected pollen to isolating. This assertion might seem like an overstatement. However, by the conclusion of this presentation, you will comprehend that it is, if anything, a conservative assessment. The reality of what is occurring within that unassuming container transcends the scope suggested by the title of this video. The agricultural innovators from Korea who pioneered this approach did not merely stumble upon an economical nutrient supplement. They effectively deciphered a 400-million-year-old dialogue between the microbial inhabitants of forests and the root systems of plants. This interaction triggers the activation of nearly 1,000 genetic pathways within your tomato plants, your rose bushes, your blueberry plants, and your raised garden beds, the instant these beneficial microorganisms come into contact with the soil. In the ensuing 18 minutes, you will depart with the precise formula, the guidelines for dilution, the scientific rationale behind its efficacy at a molecular level, and the solution to the singular enigma that eluded American researchers for 4 decades. Please, make yourself comfortable. Pour yourself a warm beverage. This information warrants your undivided attention. 1. The 1979 crystalline compound that revolutionized botanical science. Allow me to commence with an account that is largely unknown outside the specialized field of plant biology. In the year 1979, within a tranquil research facility in Beltsville, Maryland, a collective of plant chemists affiliated with the United States Department of Agriculture finally succeeded in isolating 4 milligrams of an exceptionally minute crystal. 4 milligrams. This quantity is less than the mass of a single grain of rice. To obtain these 4 milligrams, they had processed more than 500 pounds, equivalent to 227 kilograms, of rapeseed pollen meticulously gathered by honeybees from vast agricultural expanses. This undertaking consumed the better part of a decade and necessitated pilot-scale solvent extraction procedures, the collaborative efforts of multiple specialized USDA laboratories, and a level of meticulous industrial chemistry that is rarely funded in contemporary times. When the crystal finally achieved a sufficient degree of purity for examination through X-ray crystallography, the scientific community collectively held its breath for a moment. The substance they had isolated was identified as a steroid, a genuine steroidal hormone, possessing the same fundamental molecular framework as the steroidal hormones circulating within your own physiology. For 2.5 centuries of modern botanical study, it had been inconceivable that plants synthesized actual steroids. The research team published their findings in the prestigious journal Nature during that same year, bestowing upon the compound the designation brasinolide. Presently, brasinolide and its related compounds, collectively referred to as brasinosteroids, are acknowledged as the 6th primary class of plant hormones. They are a standard inclusion in all contemporary plant biology textbooks, enumerated alongside auxin, gibberellin, cytokinin, ethylene, and abscisic acid. The trade publications of that era bestowed upon brasinolide a memorable epithet that has endured: the mastermind steroid. You are about to discover the compelling reasons behind this designation. In 2010, 2 scientists from the Carnegie Institution for Science, Dr. Yu Sun and Dr. Ji-Young Wang, disseminated a seminal paper in the journal Developmental Cell. They charted, for the very first time in history, the precise function of brasinosteroid within a plant cell. The revelations were astonishing. They identified a master regulatory protein known as BZR1, which, upon activation by brasinosteroid, directly governs the activity of 953 distinct genes within the plant's genetic makeup. Take a moment to absorb the magnitude of this figure: nearly 1,000 genes. These are genes that dictate cell elongation, genes responsible for the transport of vascular fluids, genes that orchestrate the plant's immune responses against fungal pathogens, genes that determine whether a tomato will succumb to cracking under intense heat or endure a sudden frost. The keys to virtually every aspect of plant vitality are controlled by this single hormonal signal. And the title of this video assures you that you are on the verge of cultivating this vital signal in virtually limitless quantities from a container on your patio. This assurance is not a marketing ploy. It is a reflection of biological reality. 2. The Korean farmer who perceived what the USDA overlooked. Now, permit me to transport you 10,000 miles from that Maryland laboratory to a modest mountain hamlet in South Korea during the 1960s. A young farmer named Cho Han-kyu had just received a glossy promotional booklet from a representative of a Western chemical corporation. The brochure promised extraordinary crop yields, contingent upon Cho's adoption of their novel synthetic fertilizers. Numerous individuals in his community made the transition. For 2 growing seasons, the harvests were phenomenal, with rice panicles heavier than any seen by the village elders in a generation. However, by the 5th year, the soil that had sustained those families for centuries began to exhibit signs of deterioration. Earthworms disappeared. The characteristic aroma of moist spring soil, an olfactory memory ingrained in every farmer's child, vanished. Plants grew weaker, and susceptibility to diseases escalated. Each subsequent year, the farmers found themselves compelled to purchase escalating quantities of chemicals, simply to maintain the yields they had once obtained without cost. Cho Han-kyu steadfastly refused to participate in this trend. Instead, he ventured into the undisturbed woodland adjacent to his village. He knelt, gently pushed aside the uppermost layer of fallen foliage, and collected a handful of the dark, friable earth beneath. He held it in his palm, contemplating it for an extended period. No human had ever introduced a single drop of synthetic fertilizer to that forest floor. Yet the majestic oak and pine trees had stood for centuries, generating their own inherent fertility from nothing more than rainfall, sunlight, decaying leaves, and the patient activity of unseen organisms within the soil. He posed the question that profoundly altered his life, and ultimately ours. If the forest can cultivate itself without external input, why cannot a farm achieve the same? For the ensuing 40 years, Cho Han-kyu and his son, Dr. Cho Young-sang, meticulously explored methods to capture that intrinsic forest biology and integrate it into cultivated agricultural lands. In 2002, they established the Jadam Institute. Jadam, in Korean, signifies individuals who emulate nature. According to Dr. Cho's own documented figures, conventional chemical agriculture incurs an expense of approximately $5,000 per acre annually for American producers. The complete Jadam system, utilizing ingredients readily available in your kitchen and the nearest wooded area, reduces this cost to approximately $100 per acre. Reconsider this comparison. $5,000 versus $100, yielding results that peer-reviewed Korean and American researchers now describe as comparable to, and in certain aspects, superior to, conventional crop yields. The preeminent component of this entire system is the formulation residing on your back porch. It bears a simple Korean appellation, Jadam Microbial Solution. Most cultivators refer to it simply as JMS. 3. The reason doubles every 48 hours is, in fact, an understatement. Let me directly address the premise of this video's title. Some of you may have been drawn in by the phrase doubles every 48 hours, perceiving it as overly dramatic. Here is the scientifically accurate explanation. That phrase is not dramatic enough. A healthy soil bacterium, and we will specify the exact types shortly, exhibits a doubling time under optimal conditions ranging between 20 minutes and 2 hours. This is not an exaggeration. It is a fundamental principle of microbiology. A solitary bacillus cell, placed in an appropriate liquid medium with suitable nourishment, will bifurcate into 2 cells in approximately 20 to 30 minutes. 2 become 4, and 4 become 8. By the 10th hour, a single cell has theoretically produced over 1 billion progeny. Naturally, no container on Earth can replicate these ideal laboratory conditions, and the rate of multiplication decelerates as resources become limited. However, the fundamental concept remains. When you introduce a handful of forest leaf mold, containing an estimated 1 to 10 billion living microorganisms, into a vessel of mineral-rich water supplemented with a boiled potato as sustenance, you are initiating a curve of exponential growth. The total microbial population within your container does not merely double every 48 hours. It proliferates at an astounding rate. The visible foamy layer that materializes on the surface by the morning of the third day serves as the physical manifestation of this rapid expansion. And here is the crucial detail that the title cannot fully convey. Among the species present in this burgeoning population are representatives of 3 particular genera that have been extensively studied in peer-reviewed microbiology over the past 2 decades. These are Bacillus, Pseudomonas, and Azospirillum. These 3 groups, along with several others, have been confirmed in dozens of published studies in journals such as Frontiers in Plant Science, Plant and Soil, and the National Library of Medicine's PubMed database as actively synthesizing and secreting plant growth hormones as an integral part of their regular metabolic processes. Auxins, gibberellins, cytokinins, abscisic acid. 4 of the 6 principal classes of plant hormones are freely and continuously produced by the bacteria multiplying within your container. Hundreds of research papers corroborate this. The scientific consensus is unequivocal. Forest bacteria function as natural pharmacies, producing beneficial compounds for plants, and every 48 hours, that pharmacy doubles its capacity. 4. The concealed dialogue that activates your plant's steroid production. I now wish to present an even more remarkable insight, one that few gardening channels have explored. While the title of this video promises an abundance of plant steroids, their origin is more sophisticated than direct bacterial synthesis. Recent peer-reviewed research, including a 2024 study published on Science Direct examining a soil bacterium designated Bacillus endophyticus strain J13, has uncovered something extraordinary. These microorganisms do not merely deliver pre-synthesized hormones to your plants like a delivery service dropping off a package. Instead, they transmit chemical signals through the root zone. These signals, in turn, activate the plant's own brassinosteroid production machinery. The plant escalates its internal synthesis of these pivotal steroids in response to a microbial communication occurring at its root tips. Allow this concept to resonate, as it fundamentally alters our perspective. The container on your porch is not merely a fertilizer factory. It is a catalyst for awakening. You are not introducing chemical substances to your plants, nor are you even directly supplying hormones. You are introducing a population of microscopic pharmacists who, through a dialogue predating the dinosaurs, will subtly signal your tomato plant's molecular infrastructure, reminding it how to reactivate the 953 genes it already possesses but has suppressed under the chemical regimen of conventional gardening practices. The plant itself undertakes the process of synthesizing its own steroids. The microbes serve as the signaling agents. You, in your backyard with a 5-gallon container, are the host who has convened this assembly. And because you can prepare a fresh batch every week, every month, every season for the remainder of your gardening endeavors, the supply of these steroid-signaling microbes is, in practical terms, inexhaustible. The title is not a marketing gimmick. It is an accurate description of how this biological process functions. Nature has sustained this precise biological pharmacy in undisturbed forests for 400 million years, long before the advent of any chemical company. Master Cho was the first to devise a method for harnessing it, and the formulation is so straightforward that I will now guide you through its creation from beginning to end. Free Bucket Blueprint, your take-home guide. Before we delve into the recipe itself, for which I intend to provide every essential detail, please take 30 seconds to pause. This will enable you to acquire a tangible resource from this video. A QR code is currently displayed on your screen. Activate the camera application on your smartphone, position it over the code, and tap the link that appears. This link will instantly initiate the download of a complimentary PDF titled The Jadam Bucket Blueprint. Within this document, you will find a comprehensive dilution chart for every type of plant in your garden, a month-by-month application schedule tailored to your specific American growing zone, a troubleshooting guide for addressing issues such as an unusual odor from your container or a failure of the foam to form, a printable shopping list for the 5 required ingredients, and a quick reference card that you can affix to the interior of your garage door, thereby eliminating the need to memorize any numerical values. This blueprint is entirely free of charge. No email subscription is required, and there are no hidden conditions. It is yours to retain, to print, and to share with any gardening associate who could benefit from this assistance. If you are viewing this on a smaller display or currently have your hands occupied, the identical link is prominently featured at the very apex of the description section located below this video. Please take 10 seconds to access the blueprint and then rejoin me. The subsequent segment is where we will actually assemble the container together. 5. The 5-ingredient recipe that costs less than $2. The entirety of this formulation can be written on the back of an envelope. You will require precisely 5 items, and I would venture to guess that you already possess 3 of them in your kitchen at this very moment. The first item is a 5-gallon plastic bucket, not one made of metal. The rationale behind this choice is more significant than commonly understood. Galvanized steel buckets can leach trace amounts of zinc and iron ions that can disrupt the delicate microbial community you are about to cultivate. Opt for a clean, food-grade plastic container. These are available at any hardware store for $3 or $4. A thoroughly rinsed, repurposed paint bucket can also serve the purpose, as can a scrubbed-out clean cat litter bucket. Plastic is ideal. Stainless steel is an acceptable alternative in a pinch. Galvanized metal is strictly prohibited. The second item is 4 gallons of non-chlorinated water. This is a critical component. The chlorine and chloramine present in most American municipal tap water are specifically designed to eradicate microorganisms and will undoubtedly eliminate the very microbes you are endeavoring to propagate. You have 2 straightforward options. You can utilize rainwater